Okay. Hello, everyone. Thank you so much for joining us and welcome to the 5th edition of the GSU MalariaGEN Journal Club monthly series for 2025. And today's webinar is a parasite-focused one, which is going to reference Dr. Margaux Lefebvre's recent paper titled Genomic Exploration of the Journey of Plasmodium vivax in Latin America. My name is Babalo Chauke, for those of you that do not know me, and I'm a training coordinator at the GSU, working with an awesome team of people, some of whom are in the room, to organize this monthly event. So thank you so much to all of you for joining us. Just a few ground rules. Please stay on mute so that you don't disturb our speaker. If you want to ask a question, you can type it in the chat box or raise your hand later on, and we will allow you to speak at the end when you have Q&A so that Margaux can answer your questions. The Journal Club is being recorded and will be available on our YouTube channel later today, the MalariaGEN YouTube channel, so please do check it out, and you can share the recording with your colleagues, and you can also rewatch for yourself to engage with the presentation once more. Without further ado, I would like to introduce our speaker for today, Dr. Margaux Lefebvre, who graduated in 2024 and is quite excited to give today's Journal Club, and was really generous and kind when I invited her to give this presentation. So thank you so much, Dr. Lefebvre, for agreeing to give our May 2025 Journal Club. The title of the talk, as you can see on the screen, is Genomic Insights into the Evolution of Plasmodium vivax and the Emergence of Plasmodium simium in Latin America. Just a short bio so that you have a clue and an idea of who Dr. Lefebvre is. Dr. Lefebvre's research explores the population genomics of pathogens with a focus on their invasion dynamics and adaptation to new environments. In 2024, she completed her PhD at the MalariaGEN and the University of Montpellier in France, where she investigated the origin as well as adaptive evolution of Plasmodium falciparum and Plasmodium vivax in Latin America. Dr. Margaux is currently a postdoctoral fellow at the Max Planck Institute of Evolutionary Anthropology in Germany. And she's quite excited to give today's presentation, as I said. So we will allow you to ask questions at the end. So please do keep those questions, either tap them while she's presenting or be ready to raise your hand at the end. Without further ado, Dr. Margaux, thank you so much for joining us. You may go ahead and give your presentation.
Thank you very much for the introduction and for the invitation for this talk. So I will present you part of my PhD thesis work on Plasmodium vivax and Plasmodium simium. And maybe you don't know what is Plasmodium vivax. Plasmodium vivax is a parasite that is unicellular from the Apicomplexa, and it has two mandatory hosts, so human and mosquitoes, from the genera Anopheles. It is well known because during the blood phase, it causes what we call malaria, that is a disease that can kill people. And it's one of the two main human malaria agents with Plasmodium falciparum. Plasmodium vivax is the dominant malarial parasite in most intertropical countries outside of sub-Saharan Africa, where it's Plasmodium falciparum that is a dominant parasite. And it's mainly present in Asia and in Latin America. To focus a little bit more on Latin America, in the last few years, malaria caused approximately 500,000 cases, and 70% of those cases were caused by Plasmodium vivax. And as you can see on the map, the most affected countries are Venezuela, Brazil, and Colombia, so mostly in the Amazon region. And in the literature, it's a consensus that it will be the last continent to be invaded by Plasmodium vivax. But there is 3 invasion hypotheses. The first one will be an Asian or Oceanian origin, via what we call the reverse Kon-Tiki route, that is across the Pacific Ocean. And it remains mainly on antibodies detected in mummies from the pre-Columbian period. The second hypothesis is more close to the scenario that we think about the arrival of Plasmodium falciparum, the other parasite species in Latin America during the transatlantic slave trade from West and Central Africa. And it's based on work on mitochondrial DNA and microsatellites marker. And more recently in the literature, a new hypothesis arose about an European origin during the European colonization in Latin America after the arrival of Christopher Columbus. And it lays on an ancient DNA sample that is called Ebro that comes from microscope slides from Spain, dating from the 40s. But during my PhD thesis, we really want to see how did Plasmodium vivax arrive in Latin America, if we could see the differences between the three hypotheses. For that, we gather Plasmodium vivax samples from around the world. So we had more than 600 Plasmodium vivax isolates and 2 Plasmodium vivax-like isolates for a node group. And we had more than 100 newly sequenced samples for my PhD. After filtering everything, we had more than 1 million high-quality SNPs from the whole nuclear genome. And we tried to have genetic materials from almost all potential source population identified in the literature. So the one from Oceania for the hypothesis 1 from West Africa for hypothesis 2, and the sample from Spain, Ebro, for hypothesis 3. The first thing we wanted to see is, of course, the genetic structure of the worldwide Plasmodium vivax. For that, we infer the maximum likelihood tree of individuals. And we see 4 clusters on the tree. The first one is composed of East Asia, Southeast Asia, Oceania, and Bangladesh. Here, the second one, you can see the Latin American sample with Central America here in bright blue. The third one is with Middle East and South Asian samples. And the last one is African cluster with the West African diversity that connect in the East Africa diversity. We can see here that we have a well-supported American cluster. So it suggests that maybe we could have only one introduction in America. Also, we see that the European sample, Ebro, is at the base of the American cluster and is close to a Brazilian sample here. So maybe it's a piece of evidence that it could be an European origin. We wanted to go a little bit further with what we call ancestry plot here on the map. And it consists of the shared ancestry among the population. So the best number of cluster was 4, the same 4 cluster that we had for the tree. So Latin America, Africa, South Asia, and Middle East, and Oceania and the rest of Asia. But we wanted to see the substructure of each part of the world. And we could see the substructure in the Americas only at 9 clusters. Here we see that we have a first cluster that is comprised of Central American population and Colombia, and another one that is composed of countries from the Amazon basin, and the 3 other populations seems to be unmixed between them. Also, what is striking is that we see no ancestry shared with Asia or Oceania that will be seen in pink or purple here. But we see a lot of ancestry shared between Ebro and American population. If we see here that actually the Ebro sample have approximately more than half that is blue, like Latin America. Also, there is a lot of shared ancestry between West Africa and South Asia, sorry, but also with Americas, with green and black, dark red. So it suggests that we could have an African origin or European origin. We wanted to see a little bit further if we could detect any admixture between our population or even between populations. So for that, we did population networks and mixture bias. And it is inferred based on the shared drift between the population, and it allows admixture events. In this case, we have two admixture events here and here. And what is striking is that we don't have any admixture event with the American population and within American populations. And we still have one well-supported American cluster. So it still suggests that we get only one introduction. We still get the same substructure in Latin America as in before. So the Central American population with Colombia and the Amazon basin population together. And Ebro is still at the base of the American cluster, but it's not well supported. So it still suggests that maybe it could have a European origin. To get a little bit further with the arrival in Latin America impact Plasmodium vivax, we wanted to see the demographic history. For that, we use coalescence, that is the gene genealogy, and with that we can reconstruct population size from present to the past. Here you see the population size of all the population without American population. And if we compare the American population to the other population, we see that the effective size is lower, and that could be a sign of recent colonization. Also, we see a recent diversification in the American population with all the population that develops at the same time, with all the population size that divide and split. And it still indicates that we could have one unique origin. After, we wanted to see the divergence time between American population and other population. So we can see the divergence time when the two coalescence rate are splitting, sorry. And for between American and Asian population, we have more than 1,000 years ago that we have the splitting. Between Latin America and West Africa, the divergence time would be roughly between 300 and 1,000 years ago. And it's way more recent between Latin America and Europe because it could be between 100 and 300 years ago. Here we have the time when Ebro was sampled. That's why we don't have data after. So at this time of the study, we wanted to see which hypothesis we considered. So we get rid of the first hypothesis about the Asian or Oceanian origin of Plasmodium vivax in Latin America. Actually, there is no ancestry shared, and the divergence time are more ancient with Asian populations and other population. So we would think that the introduction of Plasmodium vivax could date between the 16th and the 19th century. So during the European colonization or the transatlantic trade. And so it could suggest that the European and or African population could be the origin for Plasmodium vivax. Of course, we wanted to test which hypothesis it could be. So we tested 12 different colonization scenario. Some with a European origin only, some with a West African origin only, and a lot with admixed origins. And with those 12 scenarios, we simulated 20,000 simulation per scenario with the ABC, and we tried to determine the best scenario with random forest. When we see the percentage of vote for each scenario that which one could reproduce the best or real data, we have three outliers, so that could be the three best scenario with scenario 6, that is the one that is the best, scenario 4, and scenario 8. Scenario 6 and scenario 8 are very close because we see that both of them have an admixed origin for Latin America here. And Europe could be one of the source here. we see in yellow. And also we have a non-supplet population that is involved here in light blue. For the scenario 4, Ebro could be a migrant of American population, and we could have an African origin only. But the best one was still scenario 6. So we try to infer the parameters on this scenario with more than 100,000 simulations. And one thing that is striking is that we have a trifurcation between European population, West African, and the unsupplied population. And if we see more closely to scenario 8, actually, with the same parameter, it will be the same than scenario 6. So that's why also it has so much support. Also, we see that the main source of American parasite is Europe, here with approximately 60% of impute. Also, it will be during the second half of the 19th century. And maybe you don't know that during the second half of the 19th century is one of the most important recent time migration for humans, with the post-colonial migration, when more than 10 million Europeans went to South America. So we were thinking that at this time, Plasmodium vivax arrived in Latin America. At least that's what we were thinking when I was writing this paper. But when I just submitted it, new data arrived and was published in Nature, and ancient data from Plasmodium vivax, many from Europe, and one from Peru, Latin America. And this Peruvian sample is dated before the 19th century, even like at the peri-contact period with European. And even more like this sample really cluster with the American diversity. So it's really clear that it takes part of the American diversity and it's not an outlier. So we think that maybe we could have two waves of introduction, one during the European colonization at the 16th century, and maybe a second one during the post-colonial migration with the 18th century. And we still have to see if there is any influence from the West Africa, because they couldn't test in the Megan paper because of they didn't have data from this part of the world. If we see my scenario, it makes sense because at the 16th century here, we have a first arrival in Latin America that could be the unsupplied population from us. And after a new gene flow of European sample, isolate arrive, and during the post-colonial migration, and that admixed and give nowadays Latin American diversity. So for this first part, the take-home message would be that we have two subgroups in Latin America with mixed population. There is no South Asian or Oceanian origins. We confirm the European origins with possibly two waves of migration, with the European colonization at the 16th century and the post-colonial migration at the 19th century. And we had a more complicated history than we expected. So it's published in PLOS Pathogens if you want to read it. And it even gets a little bit more complicated with the emergence of Plasmodium simium in Latin America. Maybe you don't know Plasmodium simium, it's a little bit less known than Plasmodium vivax. It's a case of zoonotic malaria. It's genetically close to Plasmodium vivax. And it's said to be a different species based on morphological traits in the erythrocytic stage. And it's called anthropozoonosis because it went from from human Plasmodium vivax to animal monkeys to become Plasmodium simium. And it affects nowadays several species of American monkeys. We have some examples here. But it's also called a zoonosis because Plasmodium simium, after jumping to monkeys, came back to human with an example of an epidemic in Rio de Janeiro in Brazil 10 years ago. But actually, a little is known about the evolutionary history of this pathogen. For nowadays, we think that it has a limited geography area in the Sao Paulo and Rio de Janeiro states in Brazil, mainly. But some infection in monkeys in Colombia and Costa Rica were couldn't make the difference between Plasmodium simium and Plasmodium vivax. Also, the history of the OGEMs is not well known because maybe you don't know that, but Plasmodium simium is genetically close to Plasmodium vivax, but actually it's close to Mexican Plasmodium vivax. And it's really strange because we only confirm cases in Brazil. And we don't know how it can happen that actually it's genetically close to Mexican Plasmodium vivax. So for that, we wanted to screen by PCR Plasmodium infection in monkeys. We got more than 700 samples from 20 monkey species from all South America, over 5 countries. And surprisingly, we didn't have any Plasmodium simium sample, but we got 3 samples from Plasmodium vivax in Colombia. Of course, with those samples, we wanted to see where does it lie in the Plasmodium vivax diversity. Even more that it's the first observation of Plasmodium vivax in American monkeys, and it could be a sign of transfer of Plasmodium vivax to new host again after Plasmodium simium. And maybe it could be a new independent OGEM. So that's why we gather Plasmodium vivax and Plasmodium samples from the literature with more than 400 samples. We had Plasmodium simium samples from monkeys and for human, and the same for Plasmodium vivax. So first, with the population structure on the PCA. On the PCA, the closer are genetically samples, the closer they are on the PCA, and we see that we still get the 4 genetic cluster in human Plasmodium vivax, with the Latin America, the African, the Indian and Afghanistan here, and the Asian here. And Plasmodium simium is a cluster that is close to human Plasmodium vivax from Latin America here, and it's already well known in the literature. For the Colombian sample that we just sequenced from monkeys, we see that actually it's close to human Plasmodium vivax from Asia, so it's a little bit surprising. And also in the literature, there is 1 sample from monkeys that is a Plasmodium vivax actually, and it's close to African Plasmodium vivax. Unfortunately, we couldn't go a little bit further with the Colombian samples because they were low coverage and degraded, so it's a little bit sad. But we would focus on Plasmodium simium because, as I show you, there is human and monkey infections, and we wanted to see if within Plasmodium simium we had any population structure because of that, or differentiation. So first we did an ancestry plot here, and we see that we have 2 distinct groups in Plasmodium simium, but we still have gene swap between the cluster with some unmixed individuals here and here. So we have the RG cluster here, that is in light blue, and here, that gets human and simian host, and the SP cluster here, where it has only human host. We wanted to see if we could see any admixture event with those population or cluster from Plasmodium simium with other American population or even outside of America. So we still use the population network from admixture bias with the admixture event. And we see that Plasmodium simium is a well-supported monophyletic group here. And it connects to the Mexican strain of Plasmodium vivax. As I told you, it's expected. It's what we see in the literature, even if it doesn't make sense at this time. And the Brazilian Plasmodium vivax from monkey would be the result of interbreeding between East African and vivax and Plasmodium simium, with approximately 40% from Plasmodium simium. So that's a lot. It will be maybe a result of hybridation. We wanted also to see if Plasmodium simium had any impact during the host switch on the demographic history. So we still use the coalescence, and we still get the effective population size from present to the past. And we see that we have a low population size of Plasmodium simium here compared to the other one in gray, population of Plasmodium vivax. So it suggests that we could have maybe a bottleneck during the host switch. But we wanted to go a little bit further and see the isolation dynamics between Plasmodium vivax and Plasmodium simium. And for that, we use MSMC-EM with a divergence time that is when the interpopulation coalescence rate here drop below 50%. And here we have the migration rate in years. So here, for example, we have the divergence between Brazilian and Mexican Plasmodium vivax. Here it could be approximately at 30 years ago. And we see a pulse of migration 100 and 200 years ago, approximately. We did that for Plasmodium simium and American Plasmodium vivax cluster. And we get divergence time that was between 10 and 30 years ago, approximately. So it's a sign that the host jump will be very recent. When we compare the migration time between Brazil and Mexico and migration rate, sorry, and between the RG cluster from Plasmodium simium and Mexico, we see that we have the samples at the same time. So it could suggest that actually Plasmodium simium could emerge from Mexican Plasmodium vivax, which migrated to Brazil approximately 100 years ago. And it's in Brazil that we could have the transfer from human to monkeys. I tried to see a little bit in the history of human at this time if I can see any pattern of migration. And the only thing that I found would be the end of American Civil War, when we had the migration of several thousand Confederates from the southern United States to Latin America, to Mexico and Brazil. And we could think that maybe the southern United States could be genetically close to Mexican Plasmodium vivax at this time. For the second Plasmodium simium cluster, that is called SP, we try also to have the migration with other cluster. One thing that is important is that we have a migration here between Brazil, Plasmodium vivax and Plasmodium simium. That is approximately 30 to 60 years ago. And no or very few genetic flows between Mexican Plasmodium vivax and SP cluster Plasmodium simium. Also, between the two clusters of Plasmodium simium, we have a recent and significant gene flow, and even more that actually they didn't have diverge yet because they didn't drop below 50%. So we could think that maybe we get a second host jump from the SP cluster, maybe independent in Brazil, or maybe it's emerged from the RG cluster. Unfortunately, we don't have enough data to say that. Also, we wanted to see if by any chance we could get some adaptation signal, and if it could be linked to the adaptation to new hosts. So of course we were thinking about the monkeys, but also humans, because it came back to humans. So for that, we genome scanned the whole core genome to seek for positive selection with some tools from haplotype length, coalescence rate, or differentiation between the population with FST. And we found 73 genes with significant SNP and regions that could be evidence of positive selection. The ones that are remarkable, we have RBP2A and RBP1B, that is well known to be important to bind to the red blood cells, either for monkeys or for humans. And also gamma, that is well known to be important to bind to human erythrocytes. We also got some genes, CMRP1 and PAT, that are essential during the mosquito phase of the life cycle of Plasmodium. And also we found no treatment-resistant genes in Plasmodium simium. So the main take-home message for that is that we discover for the first time the natural Plasmodium vivax infection in monkeys, that we really was sure that it was Plasmodium vivax. Also, it gives new question. Maybe there was several independent host change. Can it be linked to human migration or animal trade from Asia to Latin America? There is still a lot of things to do about that. Also, we saw an hybrid and genetic flow between Plasmodium simium and Plasmodium vivax, and it questions the species status. And it's really important because it can have an impact on the malaria management and the control, because monkeys can be a reservoir, and it has to be taken in account. Also, for the evolutionary history, we found that the host transfer would be recent, with the host germ from Mexican Plasmodium vivax to Brazilian Plasmodium simium approximately 100 and 200 years ago, with two genetic groups within Plasmodium simium that could be linked to a second independent host germ in Brazil, but it really has to be confirmed. And we found some host adaptation evidence to mosquitoes and primates in Plasmodium simium. To go a little bit further on those questions, I think it will be important to gather ancient or modern data from Europe, USA, and West Africa to really better understand the history of Plasmodium vivax and Plasmodium simium. For Plasmodium vivax, we really need West African sample and Europe sample to get a clearer message about how it invites Latin America. And for Plasmodium simium, I think that samples from USA or even the Caribbean could be important to better understand the migration from North America to South America. Also, it will be important to investigate the prevalence and distribution of Plasmodium vivax and Plasmodium simium in non-human primates across Latin America, because we just tried to see if we get some, and actually we found Plasmodium vivax in monkeys. It was not expected. So maybe there is even more monkeys that were infected naturally to Plasmodium vivax, and it will be important to test it. Also, it will be, to go further, it will be interesting to apply high-depth sequencing and broaden the sample collection for Plasmodium simium. For example, we could go very far with the Plasmodium simium from Plasmodium vivax, sorry, also from monkeys from Colombia. And also we were a little bit limited with Plasmodium simium samples. Thank you everyone for your attention and listening to my talk. Of course, I would like to thank my supervisor for my PhD thesis, Michael Fontaine, Franck Pugnol, and Virginie Rojon. And of course, all of our colleagues from South America and everywhere else in the world to make this kind of study possible. And if you have any question, I'm here, or even you can contact me via email if you want.
Okay, Dr. Lefebvre, brilliant presentation, really well explained, well paced, overall fantastic. So thank you so much for your presentation. It's now time for Q&A as well as discussions. I think you touched on a very important topic, which I'm not going to get into now because I'm going to allow people to ask questions. But I think the influence of politics and history on science, because we cannot talk about malaria management without looking into migration, for instance, as well. So thank you so much for this quite eruditing and educational presentation. My question, before I let other people ask questions, and I see there's already a question in the chat box, is on the monkeys being reservoirs of Plasmodium vivax. And yeah, just because I think at this point it's still a hypothesis or rather a theory, what do you think the impact is going to be on malaria management or eradicating malaria if monkeys are in fact reservoirs of Plasmodium vivax?
I think the thing that is important is first to not kill the monkeys, because it's a thing that sometimes some people do, and because they are threatened for most of them. But I think it will be important that maybe to restrict the humans to go in place where they can, because it's not the same vector between Plasmodium vivax and Plasmodium simium. For Plasmodium simium, it's mainly sylvatic Anopheles, and for Plasmodium vivax, it's more urban one. So I think that maybe if we try to be more careful with with this kind of thing when going to sylvatic area, and to just prevent the maximum weakens transmission. And also, like I say, that they could be reservoir, but we still have also to confirm that for sure.
Okay, thank you so much, Dr. Margaux. By the way, so I'm seeing two questions which I'm going to read out now. You are allowed to raise your hand, and you can be unmuted to speak. So please do raise your hand if you do have a question as well. So from the Q&A box from Anonymous, the question is, did any of the screen monkeys have Plasmodium malariae or Brasilianium? That's from Anonymous.
Yes, we tried also to see any Plasmodium malariae and Brasilianum. Does anyone else? The thing is that we get some data from that. No, I think not. I have to be sure, to be honest, because we also have a project on Plasmodium malariae and Brasilianum, but it's just at the start. So I'm not sure, sorry. But we screen for that, for sure.
Yeah, no worries, Mark. I think people can also email you and contact you to ask more questions as well, so they can get the answer as well after this presentation. There's a question from my colleague, John Ferro, who's asking, are there specific genes that change when Plasmodium jumps host?
So more generally, usually it's the genes that are linked to how to invade a red blood cell or to bind to red blood cells. And more specifically, one of the main signs in Plasmodium simium is one of the RBP gene, is that actually we have a deletion in the gene for Plasmodium simium that is not present in Plasmodium vivax. But usually it's the kind of thing that we see when there is a jump. It's really like adaptation to the mammal binding to the binding to red blood cell in the mammals. But we could, like we've seen here, could have some gene from Anopheles also for the adaptation to Anopheles, to the vector, sorry. Because also if it's different species, it has also to adapt to different vector.
Thank you so much for that, Dr. Margaux. As I said, we have about 20 minutes to go before the end of the presentation. So please do ask your questions, raise your hand, or type them in the Q&A or chat box. There's a question from Loic Kojom-Foko, who's asking, what about cryptic reservoirs of Plasmodium vivax in spleen or bone marrow that represent the bulk of vivax biomass? Your findings are based on circulating Plasmodium vivax blood, question mark?
So yeah, for Plasmodium vivax in human, it comes from only blood because it comes from people that were infected. Most of them, for the new sample, most of them were travelers. And for Plasmodium simium, we got samples from different organs in, and even in the liver, where also there is a Plasmodium vivax biomass in the liver. So we think that maybe it could be also the case of Plasmodium simium, since it's close, but we are not sure also for that. But we mainly from blood.
Okay. Thanks, Dr. Margaux. There's a question from Dr. Richard Pearson, who's saying, very interesting talk, Margaux. Do you think short read sequence data, for example, Illumina, is fully sufficient for your analyses? Or do you think you would understand more if you had long read sequence data? That's from Dr. Richard Pearson. Margaux?
Yes, thank you. For the long read sequence data, the main improvement will be to have the differences in the chromosomal structure with we could see more easily if we had deletion, inversion, or those kind of thing. We didn't took care about that, but it could be interesting if we want to see that. But I think it will be even better to have the hybrid sequencing with short read to be more sure about the base that we have, and the long read to have the chromosomal structure. I think for everything that is population structure and evolutionary history, it's in general, it's not an improvement, but if we want to be more precise in the adaptation and the difference between the species, it will be an improvement and something to add.
Okay, thank you so much, Dr. Margaux. We still have time for more questions and comments. So as I said, please do raise your hand or type in the chat box. Oh yes. So there's another question from Dr. Kojom, who's asking, some of your findings suggested a possible inbreeding between Plasmodium simium as well as Plasmodium vivax. Have you ever read any crossing studies addressing this aspect? Or will you plan to address this aspect? That's from Dr. Kojom. Dr. Margaux?
Thank you very much for this question. So actually, this sample come from the literature, and it was already seen that it was very close to Plasmodium vivax in Ethiopia, without really tested if it was an hybrid or from interbreeding between the two species. But the thing is that in my study, we use different threshold for different filter, and we try to adapt with low coverage data because some data were not well covered. It's why maybe we could get a little bit more information about that. The thing that would be cool is also to find another one or other way to find hybriding between the two species. I think for that, it will be to go to the field again and find other samples, and by luck to have one. So yeah.
Okay, thank you so much, Margaux. There's a question again from John. About the sampling, how do you know if a monkey has malaria?
So about the sampling, actually for the monkey, it was not sampled at all for malaria at the first thing. That's why also we had from the Colombia, it was degraded and low coverage, because actually it was not, the sampling was not for Plasmodium sampling. And for at least Plasmodium simium, I only found in the literature one occurrence of a monkey that has malaria. But for the other Plasmodium that infect monkeys, I think they have the fever like human and things. I'm not really sure, but I think it's very close to what human could have if it's symptomatic.
Okay.
But sorry, just to follow up. So the monkeys were kind of being sampled for other reasons, or their blood was being taken for general system sampling. Okay.
Yeah, it was some for it was for the yellow fever, some for other things. And for the literature, also it depends on the literature. So that's why we didn't test if it had malaria before sampling it. Just we sampled, and by chance we try to see if we get malaria parasites.
Really interesting and exciting finding. There's a question again from Dr. Pearson. What do you think are the relative probabilities of eradicating Plasmodium vivax versus Plasmodium falciparum? Dr. Margaux?
Okay, very tricky question. The thing is that I didn't talk about it, but Plasmodium vivax is well known to have dormancy, contrary to Plasmodium falciparum. So that means that you can get infected by Plasmodium vivax, and it stays in the liver during some time, very long time sometimes, and you don't have symptoms at all. But if you get infected, or if sometimes life happened, you can get a new relapse of the parasite in your blood. The thing is that actually we already eradicate those both parasites in Europe, because as we can see, we has both parasites in Europe in ancient DNA, and it's well known that we add it in the historical records and everything. But I think it takes money, it takes public facilities, public management and things like that. I do think that Plasmodium vivax will be less easier to eradicate because of the dormancy, but Plasmodium falciparum seems to adapt quicker to the treatment than Plasmodium vivax. So could be difficult to really see, actually.
Okay. Thank you so much, Dr. Margaux. Another question from Dr. Kojom Fako. Have you included vivax isolates from the mosquito vectors in your analyses?
No, we got only isolate from primates and from humans mainly, because it's easier to take it. And also we get most of them from travelers, at least for our data, or newly sequenced vivax isolates. So of course the travelers was the human and not the mosquito, so that's why.
Okay, thank you so much. We still have about 10 minutes before the end of the webinar. And thank you so much for all your questions. I think this is quite exciting, Margaux. You can tell that people are interested from the many questions that have been asked. And I'm sure there's more. So they can email you, they can follow you on X or on LinkedIn as well. Are there any more questions from the participants? Please raise your hand or type in the Q&A or chat box. And Dr. Margaux will gladly engage with you.
I actually had another one, if that's all right, which is if you've used any of the like MalariaGEN data resources like PV4, or I know that I think PV5 is kind of going to be worked on soon.
Can you repeat? Sorry.
So there's these MalariaGEN like data resources of hundreds or thousands of whole genomes of vivax, and yeah, is that kind of the right type of data to do this analysis, or did you need something different?
No, I think it's the kind of data that we will need. The thing is always like to have a lot of data is important, but it's also important to have the right data at the right place, if I can say it like that, because of course, depending on what are your question, like, for example, we really wanted to test a West African origin scenario for Plasmodium vivax, or to test the influence of it, because we couldn't think that even if nowadays there is not a lot of Plasmodium vivax in those areas, even if it's more and more, or even more, it's less and less a consensus nowadays because of the deep negativity that it's free from Plasmodium vivax. Actually, it's not so free from Plasmodium vivax. We really wanted to test it, and I think that the thing is that I got lost, sorry. The thing is that sometimes you need to gather information from the right place to really answer your question. But of course, you always need something from everywhere in the world to see the global population structure and the global history, or even just to have odd groups or test population, anything like that.
Okay, I see John is nodding there. Thank you so much, Dr. Lefebvre. We still have about 8 minutes. You are allowed, as I said, to raise your hand or type in the Q&A box. But Margaux, in the meantime, I think you said you submitted the paper.
Yeah.
Do you know when is the preprint that's going to come out? When is the paper going to be published? Any idea?
Actually, I submitted it yesterday, so.
Okay.
But the
well, congratulations.
Thank you. The preprint is already online if you want to see. It's what is the title? Type something on Google, Plasmodium, simium, and Margaux Lefebvre, and I think you will find it. Sorry, I don't remember.
No, it's okay, Margaux. You can
own
check later.
But yeah, everything is online if you want to read it before the review and everything.
Okay, thank you so much. Looking forward to reading it and going by the paper that you published on genomic exploration of Plasmodium vivax in Latin America. I know it's going to be quite interesting. And already the findings, as you shared now, were illuminating. So thank you. 6 minutes. Any more questions? We still have time, but otherwise, as you can see on the screen, there's many ways to get in touch with Dr. Lefebvre via email, ResearchGate, or GitHub as well. But she's still on X, even though I think she's saying she's moved, because I saw your status saying you're not using it as often now. So people should contact you using these three.
I have one more, if that's all right.
Okay, go ahead.
If vivax and simium are able to jump between humans and monkeys, are we worried at all that P. falciparum, which is a lot more deadly, also has this ability to jump?
The thing is that actually Plasmodium falciparum come from the Laverania group, and Plasmodium vivax from the vivax group. So they are very different phylogenetically, and Plasmodium vivax and the Plasmodium in the vivax group are known to be more easily to jump from host or to change host. Some of zoonotic malaria case come from those cluster. And for Plasmodium falciparum, that is a Laverania group, the Laverania group is more known to really stick to one host, at least primate host, like mammal host, and not for the vectors, it's different, but for the mammal host, it's less known to jump host as easily as the other Plasmodium sample.
Okay, thank you so much. Okay. We still have time, so I will allow more questions. But is there any truly, Dr. Lefebvre, phenomenal presentation? And for you to say this was the first international one, you were fantastic. And I cannot wait to see your many more presentations as you go on to become a professor, an internationally acclaimed scientist. So really well done. This was a great presentation, and I cannot wait to rewatch the recording later and share with my friends and colleagues in the malaria space as well. So thank you so much. Are there any last comments from your side, Dr. Margaux?
No, thank you very much for everything. Thank you for welcoming me for this journal club. I was really well welcomed, so thank you very much for that. It was really cool. And thank you everyone that pay attention and that ask questions about my work. And I hope I will have other things to present later.
Okay. Merci beaucoup, Dr. Margaux Lefebvre. And thank you so much to all of you for attending today. I'll see you next month on the 24th of June with Dr. Nina White, again on parasites as well, but we'll tell you more about that later on social media and in our newsletter. So please do sign up to the MalariaGEN newsletter and follow us on social media. We are on Blue Sky, we are on X, we are on LinkedIn, we are everywhere. So please do follow us and interact with our social media. Thank you so much, everyone. And Dr. Margaux, you were brilliant. Have a good day, everyone. Goodbye.